Good afternoon, everybody. It's a great privilege to be here in such an esteemed audience. Thanks for having me. I'm Natalie Curach. I come from Macquarie University in Sydney, Australia, and I'm here really as proxy for someone you might be more familiar with, and that's Saki Pretorius, our Deputy Vice-Chancellor, who's also the lead of the Yeast 2.0 project at Macquarie. Macquarie is a young university and it prides itself on being the innovative university. It is located about 9 miles from northwest of the Sydney Harbour Bridge, and we have the luxury of space and a beautiful leafy campus. We also have a private hospital on campus, teaching hospital, and we have a population of 40,000 students. We're also co-located in a tech business park, and we have many companies also co-located on campus with us. And we have been at the forefront of synthetic biology in Australia, and this is largely driven by Saki, who is our Vice-Chancellor, which I said. So Saki brought Yeast 2.0 to Australia, to Macquarie University, and also to our partners at the Australian Wine Research Institute. And this move really was the launch pad for the Symbio initiative, which is now a priority under the university's strategic research framework. The initiative benefits from the strong executive support and the investment of millions of internal funding. And other major supporters of the initiative have been the state's Office of the Chief Scientist and also the Department of Primary Industries with strong agricultural focus, as well as Bioplatforms Australia. And my role at the university is to manage the initiative. So we built up over the last few years, since 2014, from outdated, empty laboratory space to modern facilities, and they are customised for synthetic chromosome assembly. And we now arguably have Australia's leading yeast molecular research laboratory. The university also was the first to offer a capstone course in synthetic biology in Australia, and that was the start of the pipeline of the local skill set and expertise that we are trying to build. The students which participate in this capstone subject are also the students which participate in the iGEM competition. Macquarie has also grown a critical mass. We now have 10 laboratory leads with their teams of postdocs and postgraduate students under the synthetic biology umbrella. And we have capacity now under a range of cell types and across the kingdoms, I suppose. We have a strong foundation in structural protein and protein engineering. We have capacity in viral genomes, various microbial cells and communities, and including yeast, of course, and filamentous fungi. Also chlorophytes of marine and freshwater species all the way up to higher plants. We've recently also obtained another hire, so we now also have capacity in engineering mammalian cells. So this is just about our iGEM team, because where the other Australian universities have participated in the competition over the years, Macquarie has been the most consistent since 2010, and with ever increasing success and also with ever more growing bands of undergraduate students wanting to participate. The group from 2017 won the Best Energy Project by engineering E. coli with a hydrogen gas producing gene cluster, and this project has obtained a lot of interest, and it's now morphed and is continuing and showing some real promise. Other capacity and areas of research at Macquarie University include creation of gene circuits. We have projects on C1 assimilation, protein and enzyme engineering. We do a lot in glycomics, genome engineering and synthetic chromosomes. So it commenced with Yeast 2.0. We are responsible for chromosome 14 and chromosome 16, but now also moving into other types of cells and green lineage. Yeast metabolic engineering for flavours and fragrances related to the wine industry predominantly, and biosensors for directed evolution approaches, RNA engineering to identify novel mechanisms and regulations of control for gene expression. and wholesale biosensors for horticulturally relevant abiotic factors. Macquarie University was founded with a strong foundation in proteomics, and we're also home now to the national facility of the Australian Proteome Analysis Facility, and the guys at APAF service the community nationally and internationally. They're organised into teams of based on the technology and expertise in certain techniques, and their key strengths, well, as you can see there, protein, chemistry, array technology, mass spec, and bioinformatics. They pride themselves in their multivariate data mining, software development, and machine learning capabilities. APAF is currently undergoing growth to increase capacity to meet the growing demand for phenotypic analysis of the engineered organisms and cells that are coming from the SynBio groups. And this ties in with the aspirations of Macquarie University to establish a national genome foundry facility, which we don't have yet in Australia. We have been working to gain national support for such a facility, but in the meantime, we've also been increasing automation and high throughput capacity in a more sort of piecemeal fashion, but we'd ideally like to integrate it. So this would be valuable, as you know, not just for high throughput capability, but also to ensure that our students are receiving the appropriate education and training that they will be exposed to once they reach the workforce. So that's enough about Macquarie, and it's meant to be addressing Australia as a whole. And there are many groups across Australia under the SynBio umbrella. Macquarie being notable along with the University of Queensland and Queensland University of Technology, as well as the Commonwealth Science Organisation, the CSIRO. The CSIRO in 2016 formed a future science platform in SynBio with a $13 million investment. It is led by Claudia Vickers, which some of you may have met, and she's done a good job in leveraging those funds to in creating joint appointments, fellowships and scholarships with many institutions across Australia who have research programs that align with CSIRO. The application domains of the FSP extend across foundational technologies, societal impact, chemicals and fibres, environment and biocontrol, health and medicine. Another relevant initiative in Australia is the Queensland Government's Biofutures Plan. Queensland Government is the pointy bit on the right-hand side of Australia, that's Queensland. And partly motivated by the downturning in the mining industry, the abundance of another natural resource being sugarcane has motivated Biofutures' ambitions to have a $1 billion export-orientated bioproduct sector by 2026. Aided by state government incentives, this has drawn the attention of many companies, including Amyris or Gevo from the US. But this strong push for an industrial biomanufacturing sector is also a driver for the development of policy and coordination of infrastructure to enable the foundational biological engineering research to flourish across the country. So this scheme is closely tied to the research programs at the AIBN at the University of Queensland and Queensland University of Technology, with emphasis on metabolic engineering. So all of these efforts from the ground up brings me to the national policy initiatives which are taking place. Over the last 18 months or so, there's been a great momentum in the development of national science agenda for SynBio. And there are two significant reports currently underway. First, we have the ACOLA report, which is the Australian College of Learned Academics, commissioned a horizon scanning report. And it is used to inform science policy development, and it's due for release sometime this month. And concurrently, we have the National Research Infrastructure Roadmap being formulated to determine infrastructure in science facilities, and that's due for release in August. Macquarie's been very active in making submissions to the writing of these reports, and we're very much looking forward to hearing the recommendations. And we see this as an opportunity for streamlining and integrating facilities across the country and service to reflect the increasing interrelatedness of the discipline of Symbio. We're also feeling positive. We'll align our aspirations for increased facilities and the Genome Foundry at Macquarie. I should also mention Bioplatforms Australia. They oversee the national omics facilities around the country, so they coordinate large collaborative projects spanning environment, agriculture and health, and 2 technique-based areas which are bio-nanophotonics and synthetic biology. Australia's unique biota lends itself to a lot of these big genome screening projects, for instance, the Great Barrier Reef or endangered and even extinct marsupials and mammals, as Australia does have a terrible track record in mammal extinctions. So with the lack of a national policy, researchers in the field established their own association in 2015, and that's Synthetic Biology Australasia. We've had 2 conferences, 2016, 2017, which have been great successes, and the next one is due in 2019, probably about November in Brisbane. If you're interested in coming, you've got plenty of time to organise a holiday around it and perhaps go to the Barrier Reef while there's some left. And if you can't wait that long, there is another meeting this year, the 7th International Yeast 2.0 and Synthetic Genomes Conference, being held in Sydney this year. So it's going to be an extravaganza. And if you come, I promise you'll hear great science, but you'll also have a fabulous time. And this, thank you to our yeast group, that's our core group of their names, and that's the photo of the SBA last meeting. Coincidentally, everyone standing in the shape of Australia and Sydney Harbour. Thank you very much.
Now we have Dr. Elibio Rech from Brazil, who has brought a group of scientists to GP Right, latest international country to join. He is from EMBRAPA, which is the National Institute of Science and Technology in Synthetic Biology, and he is remote.
Hello. Good afternoon to you all. Great pleasure. Thank you for the invitation. My apologies for not being able to be personally with you this afternoon. I'll try hard next time. The first slide is already in. Can you hear me? Is that right? Can you hear me? Hello? Oh, yeah, it's okay. I'll talk about how some of our studies and some of our attempts and approach and a general roadmap about some experiments we've been doing so far. Then next slide, please. The second one. What I'm going to mention to you this afternoon has been coordinated by James Birchley from University of Missouri and Bogumil from Western University and Designing Microbes. I'm going to mention quickly about 3 questions we have about genetic engineering and how we can contribute to intensify and expand and make it more precise. One of them is how to generate a precise genome as a biochassis for engineering. In fact, the genomes we have so far, most of them we cannot really use for editing because it's not enough. There is not enough precision in the sequence in order for we to be able to find and localize what we want to engineer. So one point is we've been concentrating and generating a soybean genome through precise editing. The second one is the question whether we can use the integrase to produce genetic circuits and regulate how to switch on and off genes in plants. Our pilot project have initiated in plants, but we've got some interesting results, so we expand that from mammalian cells, including tumor cells, human cells, and also animal cells. And the third one is to use the integrase to generate large sequence like a chromosome-like in order for we to regulate entire pathways. The fourth one is to develop new technologies for delivering large DNA fragments in plant cells and eukaryotic cells, in fact. I'm not going to mention more about that because Dr. Bogumil, that's probably going to be after my presentation. He will show you some details on that. The soybean genome, the assumption is what I just said, is to use predictive modeling for systems biology. We high precision genomes, and we've been developed that, and the sequence in an advanced way. We hope to have some results in the next, by the end of this year. And as I've said, the objective is to provide systems to understand and to precise control. We're going to concentrate initially in oil and protein biosynthesis because it's a high demand from the market, but it's going to be as a proof of the concept. So we include two strategies. One is the breeding that we've been using advanced lines. So once we edit this genome, we can very quick actually generate an elite event and maybe a commercial event and synthetic biology. And the roadmap, the general roadmap for the soybean genome is in the next slide, please. Is that we've been using, as I said, some advanced lines from tropical lines and that we use in Brazil. And we're going to explore all the technologies we have so far, and a very good one, more precise ones, including the nano one, which I did not describe here, but we can to use also and compare which one is going to give us, in fact, we're going to use all of them to generate and to fill in the gaps that one technology and the other technologies do not succeed to give us the information. And further on, we want to explore the connections between mutation, recombination, and polymorphism in soybeans. Next year, up to 2020, we want to explore and generate this what we call a biochassis, but as a metaphor, and to represent a more precise annotation and regulatory activity. We are also going to use deep learning to detect, well, that's the idea, to detect regulatory landscape of genetic sequences, such as promoter and sequence, in order to construct and understand the protein and oil biosynthesis. The second issue is, and the question is, are we going to be able to switch genes in plants or in eukaryotic cells on and off according using either an inducer, an external inducer, such as could be a chemical, a physical, or a biological inducer, that we can actually switch genes on and off in the exact moment that we want to do that. And to evaluate the principle and to generate a genetic circuit, we do need first to have several integrases. We have the Phi31, the XP, also that we know that works, but to generate genetic circuits, we need quite a few ones. So we decide to evaluate, otherwise you cannot actually generate a circuit. You can just switch one on and off. And we are exploring the capacity, the slide that mentioned is phage integration and excision, that's got the capacity for we to actually add attachment sites within the genome. And the next slide, I think, is for which we can either flip promoters or codifying sequences in order to create circuits and generate and switch genes on and off. There is one slide that shows the GP. I'm not sure if you are already there, but it is showing the GFP off that we use as a model. In the next slide, you see that we can flip the DNA in order within the genome and make this on and off. We've got some quite interesting results so far in plants, and as I've said, we expand that for mammalian and mammalian cells, including the fibroblasts from bovine and also tumor cells from human. And we have already evaluated more than 10 different integrases, and most of them are working. Some are interesting in plants. We have some more integrases that works unexpected, but resulted. But in mammalian cells, we have quite a few ones. If we compare with plant cells, that we have more integrases showing a very good capacity to switch genes on and off. So the roadmap is that we are finalizing the evaluation of several integrases in eukaryotic cells. By the end of this year and early 2019, we want to design and construct several genetic circuits, either for all these eukaryotic cells that I've mentioned so far. And also, we've been developing some telomere-mediated chromosome truncation technology using small chromosomes. And together, this work is mainly coordinated by James Birchley from and which, as I've said, that I'm not going to go in detail because Bogumil is going just in a few minutes to talk about that technology about large DNA fragments. This is certainly the most important demand that we have so far is not for really to generate synthesis, but also to how to manipulate this large DNA fragments and introduce and make it functional in plant and animal cells and human cells also. And we have been in collaborating with Bogumil that's got some quite nice ideas and very important technology that's probably going to be decisive for which you actually generate plants with large fragments. Oh, I think that's what I have now. Thank you so much.
Thank you very much for giving me this opportunity to speak here. I have only 6 minutes and I will present multiple projects that we are working on in Canada, so I will very briefly go on it. I am at University of Western Ontario, located in London, Ontario, Canada. I am assistant professor. So there are a number of problems, a number of specific objectives for Genome Project Right, and I listed them here. First, we have to select organisms, and in addition to mammalian cells, we're going to be working on other organisms. Then we have to improve DNA design, editing, synthesis, DNA assembly, packaging, delivery, installation. Some of the things that I highlighted in red is projects that I identified that we are already working on in Canada. And again, you know, I probably missed a lot, so those are just some of the projects, but we are addressing most of them. So the first project, as you just guys recently heard, is together with Dr. James Birchler and Dr. Elibio Rech from USA and Brazil. And this project started during some of the conversation that we have during the Genome Project Right meeting working groups. And essentially what we try to do, we try to clone very large fragments of DNA, megabase sizes, inside yeast, and then move them to plant cells via cell fusion to plant protoplasm. So we already start working on this technology, and hopefully you can regenerate full plants. You know, that's really all I'm gonna say at this project at this moment. It's gonna be all done via cell fusion, which seems to be the most promising method for moving large sizes. Another project that we are working, and actually there are multiple labs working, so mine lab and also Dr. Vincent Martin, who is in Concordia. We try to engineer not only the nuclear genome, but also be able to create designer organelle genomes, mitochondria and chloroplast. So in my lab, we are working with diatoms, and we already clone mitochondria genomes and chloroplast in yeast, also in other bacteria like E. coli. We can move them around. And the question is now, how do we actually move them to the right compartment? And you know, this is going to give us a lot of opportunities to create synthetic designer organelles. So that is another part that we're addressing. Moving forward, we are also working right now on developing new species. So yeast is great, but yeast is only really good for maintaining very low GC content DNA, unless you engineer it further. And also direct transfer of DNA from yeast is not so good. So we need to develop other hosts. One of the hosts that we are already working on is soil nitrogen-fixing bacterium, Cynorhizobium meliloti. And the reasons we chose it, because not only is it very important for agriculture, but it had those two megaplasmids that then can be removed, then can be cured. All the essential genes are moved in the main chromosomes, and now we are using those origins for storing of the large DNA. And also the DNA transfer, direct transfer via conjugation was shown already to multiple hosts, bacteria, algae, and yeast. And we are actually applying to Genome Canada to receive additional funding from this project, which is between 3 universities, McMaster's, Waterloo, and Western, all 3 located in Ontario. There is yet another host species, another organism that we want to work, and this is by Dr. Władysława Grybik and Jograt Grybik, and they work on spider mite. Spider mite is huge pest for agriculture. And you know, so people are already working. Whoever ever grew anything, you probably identify spider mites. They kill plants by forming those webs, spider webs. Now, what is interesting, they have very small genome, one of the smallest genomes, only 1 unit megabase pairs, and they are distributed into 3 holocentric chromosomes with very low GC content, which could be stored inside these. So only 90 yeast strains, and we can clone the whole genome of that and continue working with it. And in addition, we can take advantage and maybe create biotechnology by producing spider silk inside these, which is actually thinner and stronger than spider silk. Some other projects, and those projects are coming now from Concordia, that are really directed by Dr. Vincent Martin, who is here in the audience, if you have more questions about them. One of them is essentially taking and automating growing cells and editing them using microfluidics. In yet another project is creating yeast strains that containing single or multiple genes that are taken from human to create all those humanized yeast cells that can be later used for drug screening or really for discovering all kinds of functions. So this is going to be part of the design that is going to feed into creating those synthetic human chromosomes that we are talking earlier on. Yet another project is creating next generation industrial yeast strains. So it is using DNA and genes really from model and non-model yeast strains. And the goal is to take all this and creating new strains that are going to be good for growing in low pHs, growing in high temperatures, and many other conditions. But also, what would be good, that we can actually create better yeast strains for maintaining DNA. So as I mentioned, one of the problems is, for example, high GC DNA. Maybe we can create these that's going to maintain high GC DNA better. Finally, we have now a Center for Applied Synthetic Biology that was created at Concordia University. Now about 20 PIs are involved in this center, so we are really moving forward with that. And in addition, there is the very first genome foundry that was also created in the same center, and it's really operated by Dr. Vincent Martin. He's the pioneer of synthetic biology in Canada. So we are really Canada was maybe a little bit behind UK and US in synthetic biology, but we are really catching up. And really to bring the whole community of Canadian scientists and to find out who wants to work on what project, to create Canadian roadmap. We are going to be organizing the first, I went too far. We're going to be organizing the very first meeting, GP Right Canada meeting, that will take place on August 13-15, which will be part of the larger meeting that is already happening there in Montreal. And Dr. Vincent Martin and myself, you can contact us if you want to participate in this and if you want to learn more about this meeting. And I will stop here. That was mostly a little bit East Coast, and now we're going to go to West Coast.
I'm Bing-Zhi. I'm glad to be here. Yeah, I will share some information about the synthetic biology in China, especially the synthetic genomics in China. Other tools, chromosomes, Sin 7 and Sin 13 is ongoing, and these two chromosomes have been completed in synthesis, but the debugging is ongoing. And we also tried some study on application based on AC 2.0, and we designed some gene circuit to precise control of scramble in synthetic yeast, and also we pick out some key genes and in the production of lycopene, and also we pick out some key genes in the tolerance of yeast in Jimbia's lab. Yeah, and also we are trying to develop some new scramble system in the ring chromosome and also in the polyploid strands. Yeah. Synthetic biology is the future of the biotechnology. Yeah, we consider it has a very bright future, and the China government had proposed a roadmap of synthetic biology in China several years ago, and recently they also published the National Development Plan for Biotechnology. In this plan, the synthetic biology in this plan, the synthetic biology is considered as one of the disruptive technology, and based on these two plan, the roadmap and the plan, the Ministry of Science and Technology is trying to announce a national key program on synthetic biology. I will share some detailed information about this program. This is a 5-year program. In this program, the total budget is 2.3 billions, and each project in this package will be about 30 million RMB. Yeah, and this program will cover fundamental research, capacity building, and also some applications. In the fundamental research, it will include the quantity analysis and the prediction of biosystems, and also the rational design and the building of genome. And in the capacity, we were trying to develop some equipment and some key technology on the design, building, testing, and learning. As to the applications, we were trying to develop some new systems on the chemical production and also the net products, biosynthesis, and also some stress resistance of the strains and also the crops. This program will cover 4 aspects, including the artificial genome and the chassis development, and also the artificial gene path and the circuit, and also the cell factory and the consortia, and the enabling technology for synthetic biology. This is a whole task models for the 5 years. And the grant call for the first year, 2018, has been announced the draft version, a version for the suggestion. I will give some information about this grant call. This grant call will also include 4 or 5 parts. For part 1, it will include the genome synthesis, including the prokaryotic and eukaryotic genomes. And also, we will develop the different tests, including the model microorganism and also the industrial organisms, and as well as the plant tests. For part 2 is the part and the gene circuit. We will develop a big library of the different biological parts. And also we will try to develop some gene circuit for the vaccine synthesis and also some oncolytic virus synthesis. And we also design some gene circuit for the stress resistance and the breeding of crops and the strains. And the environmental pollution is also very important in synthetic biology. We will try to develop the biosensor and the degradation system of the pollutant. Also, the electrical cells is another goal. For part 3 is the cell factory and the consortia. We will try to develop the cell factory for the production of chemicals and some net products, the plant net product and also the microbial net products. Also, we will try to develop some cell factory for the C1 utilization and also biomass utilization. And we will also try to develop the new cell-free systems as the cell factory. For part 4 is enabling technology. In this part is very related with our GP right. Yeah. It's a key technology for the key technology and the equipment for the high throughput DNA synthesis. It includes the new technology for DNA synthesis to reduce the price of the DNA synthesis, and also the microarray and microfluidics systems, and some new equipment. We also we also called a grant for the ethics assessment and the popularization, include the potential ethics issues and the regulation of the biosafety and the biosecurity of synthetic biology. It also mentions the IP issues. Another is the popularization. There is part 5. This part is very special. Is some other product collaborated with the local government. Is the Shenzhen government is the richest local government, I think. They will focus on some applications. There are some medicine production and also DNA storage system, and also on some cancer therapy. And they also focused on the bioimaging and the automation system development. This program this program gave us an opportunity for the Chinese scientists to participate in the GP right. And also in China, there are quite a lot of the centers for synthetic biology. This is some major centers on the national level or local local level. It's a synthetic biology center based on Tsinghua and Peking University, and also a Shanghai Consortium for Synthetic Biology, and a National Gene Bank based on BGI Shenzhen, and also two National Cooperative Center for Synthetic Biology at Tianjin University. Besides, there are quite a lot of small centers in different universities and institutes, especially in the famous institutes at CAS. This is my acknowledgement. Thank all the SC 2.0 members and our grant from the most MOE and NSFC. They also maybe give us another new grants for GP Right. Yeah, thank you.